Okay, so in this video, we're going to cover the third important component of blood, and that is platelets. And there are 7 characteristics that are important to note about platelets. So if you look on the image to the left, you see a lot of red blood cells, and then where the arrows are pointing to the tiny purplish dots, those are platelets. So platelets are super small, very compact. They shed a lot of the cell components that are in most other cells. So let's look at some of their characteristics. So the first is that platelets are contractile. So they contain actin, myosin, as well as a protein called thrombostenin, which allow them to contract. Another is that platelets are capable of calcium storage in order to be able to contract, and this means that they have residuals of their endoplasmic reticulum and their Golgi apparatus in order to store calcium. With that as well, they are able to create ADP and ATP. They still contain mitochondria. They also are important for clot stabilization because they have a protein called fibrin stabilizing factor, which helps to stabilize and strengthen clots, which we'll see in a little bit. Another is that they contain something called cellular growth factor, and this will help stimulate growth in the injured tissues to allow for repair. They also have a coat of glycoprotein, so this allows platelets to slide off of healthy endothelium and attach to injured or rough endothelium. And the last characteristic is that they need constant replacement. Their half-life is only 8 to 12 days. So now that we've gone through the important part of platelets, we're going to talk about what happens when there's actually an injury and the events that follow. So when there's an injury to vasculature, the immediate response that happens, the very first thing is vascular constriction, and this will increase the resistance of the vessel, and that reduces blood flow, helping to maintain hemostasis, or the maintenance of blood into the cardiovascular system. So the first mechanism where you get vascular constriction is called local myogenic spasm, and that's actually where the cells themselves spontaneously spasm without the need for outside stimulation, so they don't have to have any sort of electrical input. Kind of like if you think of the cardiomyocytes in the heart, the individual cells, if it wasn't for the SA node and the AV node, they would still be able to spasm. It would just be spontaneously and randomly. So that's the same thing that can happen here. And then what initiates that myogenic spasm is direct damage to the vessel wall. The exact mechanism for how the damage triggers the spasm is unknown, but it is known that the damage happens and that's what causes a spasm. The second mechanism for vascular constriction is local autocoid factors. Autocoids are molecules that aren't hormones, but they have very brief hormone-like actions, and they act in a paracrine manner, so they act in very local, nearby tissues. They don't travel through the blood. And an example of this is something called thromboxane A2. It's a vasoconstrictor that's released by injured platelets, and we'll see that in a little bit as well. And then the third mechanism for vascular constriction is actually nerve reflexes that are initiated by pain impulses associated with the injury. So when there is damage to the vasculature, the very first thing that's going to happen is they're going to try to form a platelet plug. So when you have really small minor cuts in the vessel wall, a platelet plug can be sufficient in itself to maintain hemostasis. You actually have hundreds of thousands of very small breaks in your vasculature every day, but the platelets come along, plug up those problems, and you never know the difference. So platelets can recognize the damage to the endothelium, can change their structure, and become activated. And what you see on the left is that when you have a platelet that becomes activated, they swell. They form these pseudopod-like appendages. They also become very sticky, and the pseudopods and the stickiness allow them to clump together and make that plug. And When they're activated, they also secrete activating factors such as ADP and thromboxane A2, which help to stimulate more platelet activation and aggregation. But of course, sometimes platelets aren't enough to mediate a larger injury, and so that's where clot formation comes into play. And there's also another video that Courtney did that goes into platelet plug formation a little bit more detail, and you can see an animation with it, so make sure to check that out. So this is showing what kind of overall happens if the platelet plug isn't enough. So in the top image, you can see that you have the platelets in gray that are covering up that break in the vasculature. And if that's not enough, you get cross-linked fibrin polymers to come in and form a really strong infrastructure. And after that occurs, fibrous tissue will actually fill in the clot by the invasion of fibroblasts. And after you have that fibrous tissue laid down, the clot will actually retract in order to close and pull the damaged vessel back together. And then once the vessel is healed, then the fibrous clot dissolves because it's no longer needed. So this talks about blood clots itself. So like I said earlier, they're used to repair large damage. Coagulation is controlled by a balance of procoagulants in the blood and anticoagulants. So the reason why we don't spontaneously have random clotting, as long as everything else is healthy and normal, is because we have many more anticoagulants in the blood than procoagulants. In addition, procoagulants require activation. There's a big cascade effect to where any part of that cascade can be controlled. And so we want to shut down coagulation if it's not necessary. Most clots only take 3 to 6 minutes to form, and retraction happens after 20 to 60 minutes. The initial retraction does, but of course, this can take a little bit longer if the damage is very severe. And then after 1 to 2 weeks is when the clots get fully taken over by connective tissue via that fibroblast invasion, like I mentioned earlier. And there will be an additional video that goes into clot formation in much more detail, but I'm just going to go over briefly the main things that you should recognize. So really what helps form clots is the cross-linked fibrin, like I showed on the previous page. And forming cross-linked fibrin is actually a really complex and long process that our bodies are able to complete in a matter of seconds. So you have sort of two sets of proteins that allow this to happen. So of course you have fibrinogen, which gets cleaved into fibrin monomers or activated into fibrin monomers. And then the fibrin monomers are polymerized and cross-linked. But what activates fibrinogen to become fibrin is a protein called thrombin. And getting thrombin itself is a long process. It comes from the splitting of prothrombin. And in order to split that prothrombin, you have to have something called prothrombin activator. And we'll see on the next slide just how complicated it is to be able to get prothrombin activator. And then once thrombin forms fibrin from fibrinogen, it actually releases something called activated fibrin stabilizing factor, and that will help to stabilize the fibrin polymers. It cross-links them to make that infrastructure really strong. Something else important to note on this slide is that almost all of the coagulants are actually formed in the liver, and then they get into blood plasma, and almost all of them also require vitamin K in order to be created. So vitamin K deficiencies are a main mechanism for clotting disorders, and we'll talk about that in the jigsaw breakout when we talk about pathology. Another important thing that we'll talk about again later is that calcium is also needed for almost every step. And so a low level of calcium can also cause clotting issues. So don't get too worried. I know this is super complicated, but you don't need to know all of these steps. You don't need to know the entire mechanism behind this. because, to be honest, all of this happens at the same time. And so teasing it out is just really difficult. It's a little too much. So what's important is that you know that it's really complicated. There's a lot of ways to regulate how you get this prothrombin activator, which, if you remember, that is what is required over here in order to split prothrombin into thrombin. So overall, what's important is that there's two pathways, and the only difference between the two pathways is how they initiate this formation. So the extrinsic pathway, which happens really rapidly, is caused by tissue trauma, and the intrinsic pathway, which is more slow to happen, is initiated by blood trauma, which is when the blood is contacted with collagen. And so if you think about it, both of those things are going to happen at the same time if you have damage. And so both of these can occur at the same time. The only other really important thing to note is that there are 3 factors that are involved in both of these pathways. And so a lack of any of them will definitely cause clotting issues. And that is factor 10, factor 5, and platelet phospholipids. All of those are required in the either creation of prothrombin activator or the actual splitting of prothrombin into thrombin. And then again, like I said earlier, calcium is required for almost every step, and so a lack of calcium can really hinder the coagulation process. So make sure before you watch the video on anticoagulants that you watch the video on coagulation that Courtney created, because it'll help you visualize all of this a little bit more. That way the discussion on anticoagulants will make some more sense. And that's all I have for this section.